This is a brief guide to imaging, looking at the images, interactive images of proteins on the protein data bank. Should you wish to access this site, you can always pause the video now, and the URL is at the top. Alternatively, you can find it on page 348 of your textbook. If we enter into the search engine, Nucleosome, then immediately we are presented with these options. There's quite an array. I'm just going to scroll down, and I'm going to select nucleosome core particle. Scrolling down with the options that were given, I'm going to go for this one here. And I'm going to go into 3D view. And I'll need to scroll down a little bit so you can see this. You can see the gene sequence is being given at the top there. And then we have this structure, this image here. And this is the image from the side. So let's just rotate that by turning it like this. And what you can immediately see is in the core here are the histones, the octamer of the nucleosome, and then around the outside is the coiling of the DNA in approximately 1.65 coils, turns of the DNA molecule around the core of the protein. So protein is at the core, the nucleosome, and the DNA is around the outside like this, coiled around it. And if we look carefully, we can see the ends of the protein molecules in the inside. You perhaps can recognize these structures here, these helices. Those are the alpha helices. There's a tiny little bit of beta-plated sheeting going on in here as well. And if we track these through the open loops, we find extending out to here, for instance, if you can see where I'm pointing with the cursor, out to here. These are the so-called N-terminals. In other words, the end of the polypeptide chain that makes up the histone protein. And it's these which are methylated in the phenomena known as epigenetics. Is involved in the suppression of genes. Epigenetics is a topic that we will study to do with gene expression and how the environment can influence the expression of genes and how this can be passed on from one generation to the next. So you can continue to explore the molecule by rotating it through a number of different axes like this, and we can begin to build up an image of the nucleosome with its 1.6 turn of DNA around it.